Yeah, let's
12 degrees.
Okay.
let's do that. Let's keep our standard set
There's
at
our
8 minutes
center.
a run, and
Tilts up when you look out in the distance. Go ahead
and do the last and add a level where we plunge right in the center of the deep scattering layer based on EK60.
So
Okay, and you want to do that at 500?
Yes, I'm at 45.
Or 450?
Yeah, it looks like it would be about 500 right now.
All right.
We'll keep an eye on it throughout the next half an hour or so, but it looks like it's holding steady.
Okay,
So I
cool.
would say that the bottom end of the deep scattering layer is 500. So yeah, if we push ourselves into 500, that should be great.
Perfect. I will communicate with the
And
ROV
have we
people.
started our transect?
Yes, you have. So this is the beginning of your 1,200 meter transect,
He's coming back.
and it looks like you
Go ahead,
just
man.
had a jelly
Back in.
that should be coming over about now.
Hold.
Great.
I'll
Our
turn over the
started
narration.
off. That's awesome.
So
Fantastic.
is anybody on the line, or do we have any scientists on shore? Or here?
There are people on the line,
That's beautiful.
and he thinks that was beautiful.
I should turn this out.
Another
Okay.
one left. Different guy. Different.
Zoom in.
At least 10 minutes is timed.
Yes, it
Yeah,
will be
okay,
timed.
roger,
I'll
gotcha.
let you know our status.
Well, that's nice.
It's,
What do you think about the lights there, Roland? You want
It's a little dark out there. It's,
to get those light bars out or just rotate them in?
I don't
Great.
remember what we did the last time, but I think it was the lower swing arms
Another
were at
jelly?
least slightly deployed.
Okay.
Yeah, we had lowers deployed about
Just
halfway.
getting caught up on the log here.
Okay, you want to let him go?
Sure.
I think that's the original
Yep.
guy up top. There's a streamer just above center, just to the right of the white dot.
What about this thing?
Okay, another jelly.
So same type.
Okay, so we're looking at video today. This is video from the Deep Discover ROV. We're connected to the NOAA ship Okeanos Explorer. Another Narcomedusa coming in here. That might be
Full wide.
Okay.
an additional one. This is great.
String thing. Go ahead. Zoom in.
Similar to a
We
Narcomedusa, similar to a
shortened them,
species
but
called
it-
Echina.
So I factored in about
characterized by
2
the
or 3
collar
minutes of
underneath the
setup
swimming
time
bell.
at each depth. So we'd have to.
We don't need setup time because we're just
No?
going to stay in configuration.
Okay. Yeah. If we shorten them to 7 or 8 minutes, we could add a 6th. Yes.
All right. Zoom out.
So I pointed the upper lights on the upper bar down a little bit,
Okay.
and then put the
Actually, we want 'em
too much.
Sure.
You want them up
We
Zoom
want him out.
Or is it
straight out.
So we're transiting now at 1,200 meters,
'Cause
about
everything is
3,937
forward
feet
from
below the surface.
Okay.
And we are about 4,200 meters above the Petite Spot volcano. And I think, guys, we're looking at a
Okay.
Larvation
I
house.
want to do a 6th transect. So we're going to have to make these about 7 or 8 minutes.
Okay. That should be easy. We're just going to stay in configuration.
Okay.
My name is Mike Ford. I'm a NOAA oceanographer
All right. Zoom out.
with NOAA Fisheries, and I'm joined by 2 Midwater colleagues tonight. Amanda and Liz, why don't you please introduce yourselves?
Sure, I'm Amanda Netburn. I am a Counts Fellow with NOAA Office
Does that
of
lighting work for you,
Exploration
Roland?
and Research.
It's
And
still
my
darker than the
expertise
other
is
day,
in
but I
the Midwater
don't know
today.
why.
It might just be more particulate. I don't know.
And
Just
my
broke
name's
the blacks
Liz
a little
Shea,
bit.
and I'm the curator of mollusks at the Delaware Museum of Natural
It's gone.
History.
Never mind.
Yeah,
I'm
wasn't sure
interested
what we
in
were
all
doing.
things cephalopod.
Great, thanks. And
Hey.
I think we're also joined by Asako Matsumoto. Asako, would you like to introduce yourself?
Jelly at the top.
Yep, zoom in.
He's drifting away from you.
Okay, Asako might just be on the event log, which we use to log all our observations here along our transects.
Hey, Mike, this is Shirley Pomponi. I have absolutely no expertise in midwater fauna, but I'm joining you anyway as the biology science lead on this leg of this expedition, so
If
I can
we do 8
learn
minutes
from
at
you
each depth,
experts.
You want to zoom partial?
We
That's
are
fantastic, Shirley.
only
Great.
2
Welcome
minutes
to the
behind,
Midnight Midwater
so we can do that.
run. Midwest,
3 more minutes
midnight
at
for most
this
of us
depth.
anyway.
What's the next depth we're shooting for?
1100.
1100.
we're just a few minutes into our 1,200 meter transect. We are above the Petite Spot volcano in the Marianas Trench Marine National Monument. We've been on an expedition now. This is our 18th dive. This is our 4th Midwater exploration of the expedition. Our expedition is part of Capstone, which is a 3-year campaign to look at the science, technology, and ocean needs of Pacific monuments.
Can you just watch my light
Looks like a copepod.
Copepod?
Wow, look at that. Testament to our video quality. We're able to look at copepod. And two of the goals of the overall Capstone effort are to engage a broad suite of scientific expertise, and I think myself, my Midwater colleagues represent a broadening of the spectrum of the scientific community. Earlier today, many experts on benthic fauna explored the Petite Spot volcano for geology and benthic fauna. Saw some incredible views of sea life there.
Zoom in.
Another goal of Capstone is to look at foundational data and making it publicly accessible. Certainly in the Midwater, being one of the most unexplored realms of the ocean, we strive to make these observations available. Begin to create a baseline of the interior of the Earth's ocean.
Is that a salp chain?
Yeah, zoom in.
Is that a salp chain that we're looking at?
That's what it looked like. Cool.
Ooh.
That's weird.
Oh, we just got tangled up.
Did we?
No, he did.
Oh.
We're fine. About 1 minute left.
You know what that is?
Okay.
That's impressive. I'm not sure what that is, really.
I've never seen anything that looks like that.
Okay, keep an eye on
Wow,
it.
yeah, let's zoom in on this. This is...
It does not look.
So they'd like to zoom in on this.
I'm trying.
In between
My
the
best.
individual steroids, but it does look like something that's colonial.
Definitely.
Tilted way up. He's a little bit above me here. Yeah, he's out of my way.
Doesn't look like any Siphonophore I've ever seen.
Maybe right in front of the lights.
No, it's certainly...
Yeah,
Okay. So
Whoa, cool.
that is really strange.
So what do you think that was?
we definitely got a good still of it.
That's
so.
about it at this
I'm not sure what that was. We'll be
that?
definitely looking at that
step.
in much more detail. That's very interesting. Look like 4 members of looks like gelatinous bodies. You can see some internal
That's
morphology.
the end of our
And they
1,200
seem to be
meter
connected
transect.
All right, what's the next step?
by some sort of structure that was
You ready, Don?
very unusual.
So you think, are you leaning more towards Cnidarian than... Yeah, okay, sorry.
I didn't see any tentacles.
Right. I did not see tentacles either, which puts it more
So
toward
you're just
South
driving
Lake,
up, you're not missing sea lice.
Yeah, it's only...
Right.
I gotta go come up. I think
body form.
pulling you now. Get it back up.
Might be a juvenile something. I'm not sure.
Take yourself.
Yeah.
Actually, it's fine.
Yeah, that was cool.
Okay.
Thrusters aren't working hard. There's something serious.
Oh, there's something serious, yeah, Shelley.
Oh, goodness, when you're yeah.
Speed up a little bit.
Looking down from the Sirius image, take a look at that, Mike and Liz and Amanda. Oh, you can't see it right now. It looked like it was a jelly, though. Just make a note to look at that later.
Yep.
Okay. We'll shoot for 30.
Yeah.
There we go.
I'm done. Let's line your heading up with the ship.
Yep. Yeah, I was trying to follow you around
Sure.
there before you.
11:30. So we'll go ahead and stop at 10:80.
Yep.
So, Mike, we spoke with navigators, and they are going to be able to shorten the transects so that we can get that 500-meter transect.
That's great news, Casey. Okay, so we'll do the normal set, and then we'll do the 500 at the end.
Yep, sounds good. And it looks like it'll just be about 2 minutes off of each one will allow us to get up there in time. And you are now...
That sounds good. That's really smart.
You're approaching 1100.
Survey 1 nav. Or would you like me to, or I sort of have been announcing the start and stop of each transect. Just do that on OKEX? Alright, I'll do that.
Yeah, you can come up a little faster.
Yep. I'm starting to move up now.
$1,105 meters.
All right.
Pilot, whenever you're ready.
Let's go ahead and call it.
We're starting our 1100 meter transect now.
Zoom in. Zoom in. Hold. Zoom out. Let's go.
What was that? Was that a larvation?
Looked like a chili, but you can zoom in again. There he is.
I don't.
Oh, wow, wow. Oh, no, wait a minute. That looks different. What?
I didn't really get a good look at it.
Okay, stand by. You're going to get a better look in just a few seconds.
It won't last long.
Yeah, it won't last long.
Oh.
Oh, that's great.
Oh, yeah.
What are you thinking? Larvation?
You know what it is? Oh, there's
It seems like, I mean, you can see nests in there for filtration. I mean, it doesn't seem like other larvation houses we've seen before.
Yeah.
Yeah, it's a different structure.
Yeah, and it did, like the ones we saw the other day. Yeah, the ones we saw the other day, Amanda, look, they don't have the little, yeah, it doesn't have that little tadpole tail, or at least I can't see it.
Almost thought more doliolid-like.
Wow, gorgeous.
everything we're seeing so far is totally throwing me off.
Oops, sorry.
Really different.
Let's see, zoom out.
This is great, though.
I know.
No, I mean, that's the whole reason we're doing this. I mean, it just goes to show how much there is to learn about this environment and the animals that live there.
All right, are we ready to move on? Watch lead.
Almost have
Never seen anything
not
like that.
a larvation
Yeah.
hat. that I've seen, though, they're always the house is like a little mucus ball with the animals.
Wow, that's great.
Holy mackerel, that's really neat.
So how would an organism like that have fared in a toe? Or
Oh my gosh, yeah.
Oh,
It would have been mushed.
yeah, that's
Yeah, Shirley, you copy.
that's what we get every time we come out here with the ROV. It's just so great.
Shirley, you copy.
Nice shrimp.
Yeah.
Hold.
Still dark. I'm going to kick in a step of game.
Yep. It's down below me.
I mean, not only do we get the images of the animals completely undamaged, but the resolution of the video is so high that we can
Zoom out.
really learn much about each of the new organisms that we encounter.
Right, so you can get some information about their behavior, things that you would not normally be able to do, with the, first of all, with the mush that you might get from a toe, but being able to see these, in the water column. You know what, Mike, earlier today we saw a shrimp that was swimming with exopods on its thorax and not its pleopods, and Les Watling thought that was kind of weird. I don't know enough about these organisms to know whether it's weird or not, but we've seen quite a few of these swimming shrimp.
You see anything rolling?
Yeah. Yeah, that was beautiful.
Focus on. Flicker in the upper left corner.
So at 1,100 meters, we're just on the cusp of dipping into the mesopelagic as we move upward. So around 200 to 1,000 is typically mesopelagic. This is an area in the ocean where, for us, it is completely dark. But some of the organisms here can still sense a tiny bit of light.
Something at the top.
Below that, the bathypelagic, greater than 1,000, down to much deeper, potentially that's down to 4,000 meters, is where you begin to look at less and less perception of light by anything.
That's inside your lens. I pulled focus super quick, but it was still past focus. It's another reflection up top. It's twice I saw the mirror.
Could it be
super shiny?
So a lot of these organisms that we're seeing are going to be clear or red, which in essence is black at that depth. So they're camouflaging themselves, but others are going to be bioluminescing, which of course we can't see, with even if we turned the lights out, we would not be able to see bioluminescence with these cameras that we've got on the. I mean, they're high def, but not sensitive to very low light levels.
2 minutes left at this depth.
Something at the top right.
Where are we going then? 1,000
All right,
meters?
what?
1,000 is next.
Roger that.
Is that fuzz?
I think it's a jelly.
Zoom in. I think it's just out of focus fuzz.
Yeah. Jelly straight ahead.
Zoom in.
White. Maybe it's fuzz.
Zoom in.
No, it's something way out there.
Yeah.
Gaining up.
What are we seeing here?
Like a larvation house.
Yeah,
Okay, we can zoom out.
it does.
I agree, Mike.
Okay.
Zoom in.
To what?
That white thing there. Is that more fuzz?
Yeah, I think so.
Okay. It's these, there's trails on this monitor that make it look like it's moving.
Oh yeah. There's a pink thing at the top center.
It's
So Shirley, correct me if I'm wrong, but I think this is one of very few, if not, this might be the first set of midwater explorations by ROV in this part of the Marianas Trench.
Yep, that's correct. There have been there has been midwater exploration of deeper environments, but never in this area. And in fact, I think probably the midwater dive that we did the other night might actually be the first deep water midwater dive too.
Time to go.
Time to go.
That's great. That's great.
Ending our 1,100 meter transect now.
Okay, we are ending our 1,100 meter transect.
Okay, lights are stowed.
Right, ready to come up?
Ready to come up. Starting up.
Starting up. So 1,000 meters?
1,000 is next.
Yep, moving up to 1,000 meters.
So I should mention that this particular, doing midwater explorations at these depths and looking from into the midwater, into the interior of the ocean, we're really excited to be able to do it because this is the largest biome on the planet. This extension from 200 meters down to the very bottom is an area that's almost not at all explored, but contains most of the life on the planet. And as many of us have observed before or said on these calls, if you look at kind of life on the planet, most of it would be characterized as gelatinous animals that live in the midwaters of the ocean. So the combination of the fact that it's such a large biome and the fact that it's so unexplored, it's very exciting to be able to take some time and scan through several layers on dives like this.
So you can use these data then to complement some of the other data that you've gotten from taking actual samples from the water column, water samples that would have some of the live organisms in them, as well as the samples that you've brought up in trawls.
Absolutely. Yeah, and in many cases, it augments those data by showing animals, as we mentioned before, that are undamaged or
So, Dan, I put,
animals that are exhibiting certain types of behavior,
close,
which is a significant addition.
Seems to be holding
Looks
that.
like it held it there.
Yeah.
Yeah, so we're very excited to be able to
Did
do
you go
these
with
kind
the
of things in
normal
this
daisy
way.
chain direction, or did you do Levi's like left-handed?
I did it. This one was a righty, only because
I was
the lifters
fascinated
on
the
my
other
left
night
were
when
still worse, but,
Amanda
yes, right
Netburn
past
was
the
telling us
DSR, I
about
did
the
three, as close as I could go.
larvations, how they
And then I went
build their mucus house and
That
wiggle
seems
their
sort of low.
tadpole tail
Just a
to
little bit of
filter
slack on the
particles,
thing.
microscopic particles out,
Yeah,
and
not much.
out of the water until the house gets clogged, and then it collapses and they build another one. I mean, that was really fascinating. I'm sure a lot of the viewers were really intrigued by by that bit of life history of this animal. And there, I mean, this is one of the common larger of these planktonic forms.
It's a pretty unique way of life.
Yeah, and
in your home
right.
several times a day.
And Amanda, we saw those all the way down to 4,000 meters the other day too.
We did. Pretty amazing. I think we saw them pretty much throughout the whole water column.
We did. Okay, we're coming up on 1,000 meters. We're at 1,003 meters.
Huh?
All right, we're at 1,000 meters.
Pilot, whenever you're ready.
Let's call it.
Starting our 1,000 meter transect now.
All right.
Sorry. don't know if you can hear me stuttering over here. Bottom left.
Oh, yep. Coming back. Go ahead. Larvation. Yeah. So what are these larvating? These larvation houses.
What are they? Is that what you asked, Dan? That's what they are. They're houses.
For what kind of creature?
Yeah.
Oh, what kind of creature? Okay.
Abandoned larvation house.
Top shiny bottom
Okay.
left.
You want to tell us about what kind of creature these are? One of the questions we had is, what kind of creature is a larvation?
So larvations are pelagic tunicates. They're in the phylum Chordata, so actually not that distantly related from us. And they look like little tadpoles that build these big mucus webs to filter food out of the water. And they use their little tadpole tail to pump water through that web. And it slowly gets clogged up with all this marine snow, which we can see off in the background, these little white particles. And once it gets clogged up, they just ditch their house and build a new one. So a lot of the houses we've been seeing today look like they've probably been abandoned. They start to collapse on themselves and slowly sink down to the seafloor, become part of the marine snow themselves, and actually transport carbon down to the seafloor.
So what are all these white dots that we're seeing in the water?
Something coming center.
So these
White dots
are particles that we tend to refer to as marine snow for, I think, fairly obvious reasons. Although sometimes I think they look
Just
more
white
like
dots.
stars than snow. But these are kind of bits of decaying matter that's raining down from the surface. So these are phytoplankton, zooplankton, and other animals that have died or defecated. Create all these little bits of organic material that slowly rain down into the deep sea. Eventually, some of this might become part of the sediment, but a lot of it will be kind of fed upon by other animals or broken down by bacteria, and the nutrients released back into the water column.
Where?
And Amanda, you were explaining the other day, and you guys break in if you see anything that interesting. So don't feel bashful about interrupting. But the other day you were mentioning that they're bacteria that kind of attach to these, form, there are large clusters, and through quorum sensing, form aggregates. Yeah, there we go, copepod. Oh, there we go, copepod.
It's really incredible that we can make use of.
I know.
They move really fast.
That's a good camera.
And as they settle through the water column, they're using that kind of film that they produce until by the time they get down to the bottom, a lot of that is not bioavailable anymore. So that's one of the reasons why we don't see a whole lot of life down at the bottom and very deep water, because by the time those particles reach the sediment, there's not much on there that's nutritional value.
Like a translucent or silver.
Partial.
Great explanation.
Well, you explained it to us the other night, so I was just trying to remember what it was that you told us.
I'm impressed. You're an excellent student.
I'm learning a lot, Amanda.
Sounds like our light's not transferring as far. Serious. Doesn't have
looks like I have a lot more to learn since about half the animals we've seen so far I can't even place into a phylum.
There's a white line coming to the left. Jelly to the right.
That's not a jelly. I think they're out of focus Siphonophores particles.
When they got pink, that's
Zoom in.
when I call them a jelly.
Yeah, it's just mucus.
Full wide.
What's the next stop, Nav?
900.
Say again?
900 meters.
900.
So,
Okay. Gotcha.
Center.
Just wanted to pass along the pilot and video. Great video. Our shore-based science team is saying great video, so thank you.
That's good. Sorry, we're not seeing much at this depth. Just mucus and fuzz.
Yes, I'll second that. This is quite incredible. And I know they've had a long day, so we really appreciate this. That's all of you, Shirley.
I'm going to scoot out for just a few minutes and grab a bite to eat, but Patty Fryer is here.
Who knows nothing about critters in the water column. I'm the geology lead for this expedition, and I am a marine geology research professor at the University of Hawaii, Manoa, and a member of the Hawaii Institute of Geophysics and Planetology. And we are bringing you midwater critters. Ooh, something long and stringy. Okay, folks online, what are we looking at? We're looking at muck. M-U-C stands for muck or mucus.
Yeah, zoom out. Mucus and
You
fuzz.
better correct me because I really don't know this stuff, but it's so much fun.
Yep. Yep, Patty, this is Mike. That's mucus strands.
Okay.
And that's one of the forms of the material that we see floating around in the water column here. We've seen a few of them so far, and maybe it's just me, but I feel like we have more particles here than when we started. Maybe that'll be the trend as we move up toward 500 meters.
Oh, Mike, were you watching the dive?
That's exactly what we saw in the last dive.
Top left.
Just more and more particles as we moved up in the water column, which is, what you would expect.
Well, there's a lot of stuff here. We were surprised by how many fish there were on the seafloor. We were so deep on the benthic part of this dive.
You have 25 seconds.
Okay, I'll start
There
packing
were a
up
lot of fish.
swing arms then.
I wasn't able to tune in for that portion of it.
Oh, there's
Top
something
center.
swimming across the screen from left to right, really
Can you
fast.
focus?
No,
it's gone.
I
Zoom
was
out.
full in and full out.
That could have been a
All right,
copepod.
Pat, we are all done
Looked like
here.
it was really moving.
Okay.
Boy, yeah.
That's it.
We've seen a bunch of those.
That
next pilot in
NZ 1000 transect.
So, Mike, what are the things that
like 900 meters.
change direction really fast, like, UFOs, unidentified floating objects?
Right, yeah, those would be the copepods. We see them a lot. They are small,
Pilot's next
microscopic
transect is at
crustaceans,
900
and
meters.
they're
Lights are still ready
really
to come up when
cool
you are, pilot.
zooplankton to look at. And they do dart around and change direction frequently. They have a very high
coming up.
acceleration. They're extremely fast movers, and I think they're one of the highest accelerations you see in nature. Is those jumps that copepods make. If you look at the
29,
physics and you scale it down to their body size,
28.
it's pretty amazing.
Boy, I bet.
Oh, we just got auto depth in. I bet.
They can
Oh, going down.
move.
I'm catching up.
2. Yeah, you're catching up. Should be.
Okay, it looks like we're ascending up to our 900-meter
Delta's holding at 22.
transect and getting in position.
Can you, my upper
Yeah,
swing
looks like
arms,
we-
can you tilt them all on?
Oh, go ahead.
Upper.
Oh, go ahead.
It's gone by in the
Particularly
serious
the
view.
starboard one.
Get both of them. Starboard. Swing.
Just went in and out of the frame real quick.
Oh, yeah. Come on up.
I don't
Okay,
like
we'll
it
note
when
I
you do
missed
that.
that, unfortunately.
Yeah. I just happened to have just turned over to camera
I'm at
2 and
28.
saw
I
it.
can speed
That
up
was
just a hair here.
fortunate.
So we're going 900 now?
Oh, you're looking at the Sirius view?
Well, that's always on my mind, so that's
Yeah.
I'd like to go back and see it again.
Uh-huh.
I'm really rooting for Squids on this one.
I'm really rooting for Squid too. If I had to guess, it would have been a Cranchian
Yeah,
squid.
900. So I'll
But
stop at like
I think
Ooh.
that's asking. That's
880.
excessively hopeful, maybe.
Less than that. I mean, greater than that, like 885.
Okay.
Something like that.
Yeah. But yeah, I'll get you at that level and then.
Yeah, I mean, you can adjust your height as much as you need.
I see, I think, a copepod at the bottom of the screen. Just came up, zipped across, and zipped down below the screen again.
Yep, I see that, Patty. I think that's a great guess.
Moved very fast.
Yeah, that
Midwater Transect, but as soon as you kind of stop a little bit, you start to notice that a lot of what appears to be marine snow are actually copepods or other small zooplankton. A lot more of it is zipping around than we realize when we're moving. Pretty cool.
Oh, yeah. Fascinating critters.
Okay,
Very
so I
tiny.
think there's a south chain in camera 2.
Yeah, pilot, just let me know when you're all settled on 900 to start.
And now it's gone.
Because I have to announce it. So when you're ready.
Copy that.
I'm ready.
I have a question for you Midwater folks. You know, when I'm diving in Alvin or the Shinkai 6500 and we're going down, you know, can be almost anywhere in the water column. You see a lot of bioluminescence on some of these more gelatinous things. Are those necessarily jellyfish, or are there other midwater animals that produce bioluminescence that I might be seeing?
Well, I'll take a stab. I mean, I think there are a lot of jellyfish that do use bioluminescence. But I think, Amanda, I think there's some fish that do as well.
I'm at 900 if you want to deploy my arms again.
Yeah, a lot of the fish in the water column use bioluminescence.
We are now starting the 900 meter transect.
Turnfish, which have little photophores for small light-producing organs on them.
I copy it.
Right.
Those would be the most common that you're likely to see. But then there are also things like anglerfish or dragonfish, which have spores hanging off them. And on those ones, they actually are harboring a little sac of bacteria that produce that light.
Oh, okay.
Do we have a prevailing current here?
And there are some cephalopods that also have light-producing organs on them. One of my favorite is the family Histioteuthidae, and these are covered completely, arms, bodies, tentacle, arms, head, and body in photophores, and they're called strawberry squid is their common name, and that's because when you have one in your hand and you're looking at it on deck, it looks like a bright red strawberry covered in little seeds. And all those seeds are photophores.
Oh, that's amazing.
Nope, I got more, but it's...
well, we're on our 900 meter transect now. And there looks like a worm or something sort of squiggly in the center of the screen.
Pulling out.
What is it?
It's underneath me. Okay.
What was it?
So, co-pilot, you need to tell me. I want you to keep a fixed
So
camera
on the last
I'll have it at
that we did a couple of days ago,
let me know what's
we
going on.
saw
I mean,
a
if I
lot
got
of
a really nice,
these
pretty thing,
things that look kind of like that, except a little more small. And we got some nice zooms on it.
Sounds
and
good. Keep you at
to me,
50.
it almost looks like someone took plastic wrap and
I saw it too.
pushed it up and made a little ball out of it.
Huh.
Maybe a little bit more kind of fibers in it. It didn't look like anything I've ever seen. It looked more solid than an aggregate of marine snow, and they all really had a similar kind of structure. Some of them had a long kind of stringy tail coming off of them.
Quick one.
I have no idea what they were. It didn't look like anything I've seen before.
Full wide.
Gotta be
Oh,
quick.
that must have been exciting. We just zoomed in.
Yeah, we saw a lot of them. It was very consistent, and
Yeah, you got the ship moving decimal
today.
1 or decimal 2, or
That's what we just saw at the beginning of this trip. This 900 meter transect was the closest thing I've seen
It looks like
to
it's
that
in a
so
continuous
far.
move of
We saw it, there's
0.1
not
knots.
in
Let me double
the
check.
depths, so we weren't really deep on that dive, but we saw it quite consistently.
Okay.
Yeah, we're just in one
Really
big continuous move at 0.1.
Okay.
Sure sounds it.
Go ahead.
We have seen a couple of little things. There's something that you may be seeing shortly.
There's a Keating
Oh, what is this? This thing tumbling about, head over heels.
That's beautiful.
These are also called arrow worms. They're
looks
like an arrow.
Yeah, we saw something like this close to the bottom.
Jaws.
You're drifting off to my starboard?
They're considered to be one of the greatest predators of the plankton.
They're so tiny.
Oh, do we got the lasers?
I know. It's amazing.
We were using that before.
Yeah, it's useful.
These deeper ones are a bit more, they're a bit larger and a bit more opaque than the shallower ones. The shallower ones tend to be really transparent.
Huh.
Kind of lose them in the video.
Well, if you look at the Sirius view, the water looks much murkier here than I would have expected. Is that all the biomass in the water, or what do you think?
Top center.
Yeah, I think, small organisms, and then, also just the marine snow that's here.
Yeah.
I haven't been looking closely at the Sirius cam to kind of compare this with other sites that we've got.
Right. Well, that's not surprising. You're looking for critters in the water.
I am, but
keep an eye on my tether.
Unfortunately, don't have a full control room in my house.
Yeah, looks all right.
I'm starting to think about building one.
Moving forward enough to where...
Yeah, no, it shouldn't be a problem, but if I come back really quick, I can definitely catch it on the swing arm.
Copy that.
So when can we expect to see a fish? I know, I'm cheating.
2 minutes, pilots.
Start seeing fish. You see that really young layer
pack up swing arms in 1 minute, or do you want me to pack up swing arms?
Whenever you feel
And it's likely that is
we can do 30 seconds before. I think that's fine.
Sounds good.
I mean, we can start.
Many of the fish have swim bladders.
Yeah,
so have really high acoustic reflectance.
Yeah, I just didn't know what his timing was.
At least last I looked at the EK60, we aren't seeing much below 500 meters.
Oh, right. Oh, yeah, I see that. Yeah, we have it up on the screen here in the back row. Looks pretty good there. Yep. Yeah, the way we attracted fish on the Falcor when I was out with Jeff Drazen and his Hades buddies was to put bait on a long metal pole in front of the cameras. If we put a chicken in one of the rock boxes, do you think it'll work?
Yeah, so... I'll tell you that.
Yeah, my depth's only
But that would be cheating, right?
Meter.
So he was looking at fish on the seafloor. Is that correct, Patty?
Yes, we were. These were landers that were deployed over the stern. And, ooh, there's a jelly of some sort of gelatinous thing. What is this? It has speckles. Oh, it's cool.
Yep, that's another larvation house.
A larvation house. Wow.
Start packing up lights.
Larvation house.
Really thin. Nobody home.
Sky.
Don't know in there. So, Patty, something really interesting I've seen in the Midwater. I was working with a colleague out on Ambari on the Western Flyer. And he rigged up, it looked like a little fishing pole, but just out of
This ends the 900-meter transect.
And put a sardine on it.
Next transect is 800 meters.
A camera down there with infrared light.
Oh.
To try and catch squid, but also fish and other animals. But he was really looking for the squid. We left it out there for a day or two. We came back and nothing had touched the sardines. And really nothing had come into the view of the camera. And the big question that led us all to wonder about is whether scavengers just don't really exist in the Midwater. As well as dead animals, either become marine snow,
we're gonna 800 next.
Feed on marine snow in the water column, or if they just make it to the seafloor.
And then what's that? Is there any other?
Then we're doing 500.
Okay.
Organisms have evolved that trait in the Midwater. It was really interesting. It was surprising.
Oh, wow.
But I know on the seafloor, you put something dead on the seafloor, and there are things, there in a minute. There are hagfish and isopods and other kinds of fish.
Absolutely. Yeah, we did put. Sorry, we did put a chicken from the galley into a trap on Jim Cameron's dive to the Mariana Trench, and these guys, the amphipods, came and stripped it bare like a pack of piranhas.
Yeah. They find it so quickly.
Oh man, yeah, good sense of smell.
So I just got back from dinner.
Yeah.
Wolfed down my dinner.
How did you like the chicken?
I liked it.
We should save some for the ROV.
Forget it.
For attracting fish. We were just talking about that.
Forget it. I got the last piece.
Really?
Yes. Anyway, I just saw that you saw an arrow worm, a ketognath.
We did?
And we saw one. I don't
yeah,
know,
on the sea. I mentioned it. Yeah, on the seafloor.
Yeah, what was that, 5,500
Oh, man,
meters?
really deep.
Yeah, we saw one just hanging out.
And we just saw a larvation house.
That was above the seafloor?
Yes. Yes, it was. It was, I would say, maybe...
We got good imagery of it.
Yeah, we got really good imagery of it, Amanda. It was just above the seafloor. And I think we saw one maybe the other, I don't know, about a week ago.
Yeah, I think you're right.
Yeah, but so it wasn't the first one that we've seen.
Yeah.
I was a little stumped when I saw it. It looked to me like it might be a ketognath, and as we were like, but we're going back several decades to when I took a plankton course from Mike Reeve down at University of Miami, and it looked like a ketognath.
800
and then as we were getting closer and closer to it, there were like numerous is it a fish? Is it a
we were wondering
something? Is it whatever?
Right,
we were getting lots of
was.
comments from our shore-based scientists as well. And then
but they
finally,
identified it.
Yeah.
Said the same thing you did about it. That it's a voracious predator. Yeah.
Okay, we're getting to our 800-meter transect depth.
Predator is the word I was looking for.
785 wire stop. Which will stop.
Come in center. All right, we're at 800, starting the transect. Come on in, video.
Starting 800-meter transect now.
Okay, starting now.
Holding partial.
Go ahead. Is he moving away?
So, what is this? This looks like it's something pulsing. Mike,
pulsing.
Get ready.
Ooh,
get
yeah.
Ready, Mike. Get ready.
A circular globular. Yes, it's swimming away. It's a jelly. I think.
I'm all excited. I'm looking at all the detail I possibly can. Definitely a jelly.
Oh, cool.
It's really small.
That's all right. It'll grow.
Here it goes.
Ooh. Zooming away. Oh, look at this little red spot.
Is that a scalp with him?
Yeah, it did.
Beautiful.
There it is.
There he goes.
Yeah, little hydromedusae.
Oh, up to the top of the ROV.
Yep, that looks like some sort of rapalid, really small, but
off the bottom
oh, what is this?
Detail.
Oh, this is pulsing
Great.
too. Look at the top of it.
Siphonophore, maybe.
Oh, what is it?
Siphonophore.
Siphonophore.
Ha. Okay, what's a siphonophore?
Yeah, so siphonophores are gelatinous animals that are
little guy.
kind of all work together
From your stern to your left.
and are joined by one common cord. And you'll see the pulsing bell at the top is a nectophore, and it has converted its body for the sole purpose of propulsion. And lower on the core, you saw those little elements in the morphology. Those are organisms that dedicated their body form for things like reproduction and feeding and defense. So it's an organism that kind of works together in this common cord. So it's pretty cool.
It is indeed.
Lots of them.
I can angle
Okay,
down a little
looks
bit
like
if you
we're
want.
seeing.
Is there another one or
No,
something
it
on
looked
the
like
right?
it looked like it might be a larvation that was
The
coming up.
larvation
56.
house?
Yeah, it was a larvation house. So, Mike, what are siphonophores related to?
What are these?
Come on in tight.
They're related to hydromedusae and other jellies too, so they're the same general
I'm
group.
not sure it came out of the light, yeah.
Huh.
There's something
Sometimes these
in
things
the
get to be
screen.
enormous, like
Really?
tens.
for it.
Oh yeah, tens of meters
Fish
long.
just swam by.
Tens of meters?
Yes.
Sirius's view. And he's going-
Whoa.
Well, maybe
so
not.
The
I bet.
hydromedusae and... Oh, sorry, go ahead.
No, go ahead, Mike.
That's, wow.
The big group of them is typically called the hydrozoa,
Okay, come on in.
which would include some benthic polyps, but also the little hydromedusa, like the small one we just saw, up to the lights of Deep Discoverer, and then Siphonophores, which are the colonial animal we just talked about, or the animal with the nectophore and stuff. So that's usually that group of three types of organism in the Retigelatinus are called the hydrozoa.
Concentric rings.
Roger.
Very cool.
Do you want me to tilt back to 50 if you push out a little bit?
What are you at now? 56?
Yeah.
All right, come on in.
Okay.
Ooh, check this out.
Oh man,
Full zoom.
Oh my God, check this one out. Oh, Mike, wait till you see this.
this looks like something
Oh,
from
wow.
Star Wars. Just dripping
really
with
pretty.
tentacles.
Gorgeous.
What is
Another
that?
jelly.
I can tell you what that is.
Oh, there he goes.
That's our highlight image for today's
You
slide.
bet it is. Yeah, that's gorgeous. Look at the teeny little critter moving around there.
Yeah.
What is that teeny guy? He's trying to imitate the big
Another
one.
hydromedusa.
Is it?
I think so.
Oh, wow. It's having babies.
Yep. Some of these hydromedusae are have their young medusae grow up in under their bell.
Ah.
How much time we got, Navigator?
So in that
just under 4 minutes.
hydromedusa that we just saw, the one that had all those like vertical, are they all like were they all tentacles that looked, like those reddish?
Yeah, they were sort of
Yeah, were they all
horizontal
tentacles?
arms hanging out, and then the tentacles came down
Yeah.
from all of those.
Yep. Those
Oh,
are
there's something on the
tentacles
left.
all kind of hanging out, and at times
What
they
is
can
this?
retract them, and then at times they can let them extend fully in sort of the
Holy
two-foot
smoke, look at
predator
this.
mode.
Spectacular.
It's got immense
Looks like-
antennae. It's got gigantic
looks
long
like it has
antennae.
chopsticks.
Yeah, it's
I
just
should put chopsticks.
holy smoke.
Oh,
Antennae
wow,
are
wait till you see this.
Oh goodness, that's a beautiful image. I don't know. This is a good contender for image of the day with
I
those
know.
long antennae. Although I have to admit the jelly
That's
was
incredible.
Roger.
That's gorgeous.
Sounds good. I'll hold this tight shot for a moment.
Look at this guy.
What a great portrait. Portrait from the deep.
So there are a lot of animals in the deep sea that have
I'm going to come
really
just a touch
extended
wider.
appendages like this.
Mm-hmm.
And part of the reason is that, kind of like cat whiskers, it gives them a much greater area that they can sense. Because it is really dark down here, they're not able to use light and vision as much. So this, you can see how much that increases. What's kind of
Tether's happy,
available
but
to
it is
that,
off to
that
port, so don't
defense.
turn any further. Port.
There's some
2
I know
minutes.
of that have that kind of bending back in their
when you're happy.
antennae or suggested shape, but I'm not sure if this is one of those or not. It looks pretty different than the ones I'm familiar with. Really awesome.
Wow.
Incredible.
Something up to the top of the screen, but it's
Going off screen.
There's-
Let's get this guy.
yeah.
Oh, is that up in the upper right? Maybe another. Is that another arrow worm? I don't know.
It is. It's upside down. Yep.
Yep, but it is.
It looks like an arrow worm upside down.
Yep, it is.
Oh, yeah. Wow. Wow.
It's getting very close to you.
Big.
Yeah.
Thick.
He's gone. So hard to tell.
Yeah, I know.
Wow.
Nothing to reference.
How are we doing on time now?
35 seconds.
Okay.
There's something just about a center screen. Pretty far though.
I'll pack up the lights.
Okay, go ahead in.
Snappy.
All right, another one.
Oh, look. And this one is wiggling its tail.
Wow.
Fish.
Swing just dead.
You can see it open its mouth. You can see its eye.
Yeah.
Coming in on
Oh, this is such
wow.
a cool video.
What
Wow,
is that?
Very
Oh my
I
gosh.
come just a touch further out because
looks like a baby fish.
It does, doesn't it? Oh.
I don't see fins,
On the
but
side.
they may be really delicate.
and he's going up.
Well, I asked for a fish.
Out of
So
the
I
way.
don't know what.
Wow. That's
Think it's a
cool
fish?
shot
I
there.
don't know what kind of
Cool.
fish
There he
this
goes.
is, but this behavior
Time for us to come up.
Yep. What's
is
our next step?
That's fun.
500.
kind of
Yes,
typical of some of
500
the
meters.
deep sea predatory fish.
Ah,
That sort of
That ends our
like
800 meter
operate.
transect. Next transect is 500 meters.
How late were we shown?
27.
So it allows them to find their
Almost
predators by
30
looking for the shadows and the downwelling light from above. As Mike mentioned before, we're now in the true mesopelagic zone, so there is a little bit of light that reaches here. The fish will do that, just kind of hang upward in the water column, save their energy, and then when they see a shadow come by, they can go after their prey.
33. Oh.
That was really neat.
it
Very
was.
neat. Okay, we are moving up the water column to our final Midwater transect at, I believe, 500 meters. Is that correct?
32, 33.
That is what the pilot said.
Yes, 500 meters is the next and last transect we have.
So correct me if I'm
Next
wrong.
to
I think
last.
what we're doing now is we're entering this zone where we're really in we were at 800 meters, sort of at the bottom end of where the vertical migrators might go during the day. Now we're kind of tipping up closer to where they might venture at night. So anywhere between 800 meters, around that level, might be where some of these animals hang out during the day, and then at night they might travel up to as close to 100 meters up to the surface.
Specific things.
So that might help us describe a little bit of what we might see here. And that distance for small organisms is actually incredible. A lot of plankton do this movement. And I was looking it up, and it's the equivalent of a person taking a 25-mile run twice a day.
Does it flicker
Whoa.
to this state?
And finishing a 25-mile run in 2 hours.
Does the button stay the same color on top?
Pretty
or does it turn
speedy.
yellow?
Yeah, I mean, it really don't. It's very impressive. First of all, the distance and the scale of the migration, the number of organisms doing that every day, the distance that they travel every day, but the sheer mass of that. I mean, when you think of some of these massive animal migrations,
True.
and they all pale by comparison to what happens every single day.
That's for sure.
In the midwater. You've been hearing from Mike Ford from NOAA Fisheries. Is that right, Mike? NOAA Fisheries?
33
That's right, Shirley.
And Liz Shea from the Delaware Museum of Natural History. Amanda Netburn from NOAA's Office of Ocean Exploration and Research. We have Asako Matsumoto joining us on the chat room. She has every day for all of our dives. She's from the Chiba Institute of Technology in Japan. Thank you, Asako. Who else do we have? Debbie Blaney from Ocean Exploration and Research. And I'm not sure if there's anyone else online. I apologize if I didn't mention your name. And for many of you out there who are sharing in the excitement as we go through and explore these midwater regions, the largest biome on our planet. My name is Shirley Pomponi. I'm the biology science lead on this leg of our expedition. I'm from Harbor Branch Oceanographic Institute at Florida Atlantic University. And my sidekick
is her geology lead partner, Patty Fryer from University of Hawaii at Manoa, Hawaii Institute of Geophysics.
And we
are having a great time.
We are having a great time. And we're our very important partner is the Global Foundation for Ocean Exploration, and these are our ROV crew, pilots, navigators, co-pilots,
and videographers.
Videographers, exactly. Did I say nav? I didn't. I don't know if I mentioned nav.
You did.
And right now we have as our pilot Josh, as our co-pilot Andy. Is that you? Oh no, it's Levi.
Yeah.
Hi, Levi.
Levi.
It's Levi.
Hello there.
We have Sean. Boy, you've been bouncing all around today, Sean, haven't you? As navigator, we have Tara as our videographer right now, and we have Annie, who's sitting in the back doing video clipping, and we have Casey Cantwell, who is our most excellent expedition coordinator from the Office of Ocean Exploration and Research.
Yeah, you gotta slow down a little bit, Levi.
So for those of you who are following us at home, and I know there are many of you who are doing that, the way you've heard us communicating by phone, so we have a phone line, and scientists who have registered to participate as part of our shore-based science team call in and talk to us, and they're our phone of friends. They're the ones who are providing a lot of the information on the organisms that we're seeing.
We won't feel surface current until
the behaviors that we're observing.
Can you see any current at 500?
And we also have a chat room, so we're furiously typing notes to each other and making notes in an event log that will be archived. So we have little codes for all the things that we see. We need a code for Ketignath.
Yeah, and there is absolutely no way to go in and change your typos. So you gotta, kind of be careful here. And I'm not, so I apologize to anybody who reads this in the future. Even I can't figure out what I'm saying sometimes. But
that's why we have the codes.
Excited. Yes,
that's why, but sometimes even the codes.
Well, the descriptions of, ROC for rock, and then you describe it and you type the wrong word in. TEH is my favorite word for the.
Okay, we are moving to 500 meters, so we can place Deep Discoverer into the deep scattering layer as seen in the 70 kilohertz EK60 trace, which you are seeing, I believe, on your screen 3, video feed 3.
So people following at home can also see the scattering layer on video feed 3 in the bottom right corner. The bottom of the two images shows a rather thick gray line at about 500 meters.
Looks like that was a fish there.
28, 29. Buff out. It's okay. You getting close to me?
Well, we do in fact seem to be entering fish territory once we get close to the flyer.
Try to get it right at 30. I know, but it's bugging me. Okay. Yeah, that's my fine tune.
We are at 535 meters. We'll be up to our target depth of 500 meters in about a minute.
Okay. I want another fish. You can tell they're fish because you can see their eyes.
This is kind of the part of the midwater exploration where each one of us hopes for something massive and impressive area that we work on.
I can tell you that all of us here in the control room are thinking the exact same thing. The ROV crew was telling us,
the meter-long makes sense good.
Well, they were telling us one day towards the beginning of this leg, they actually did see a squid that was about a meter long. It's about a meter long? Really? Yeah, big. Oh, wait. How big was that, Josh? Oh, that looks like 2 meters long. Was that a fish story? Yeah, pretty big. And it was like it was very curious about the deep discover. Okay, we are coming up on 500 meters.
Going out.
Settling out.
All right, we're starting our 500 meter transect.
Oh my gosh.
Oh,
so Mike, do you know what this is?
Okay, going for full zoom.
Looks like the crystal entity from Star Trek.
There it is.
Another one of those, yeah.
Do you remember this from Puerto Rico?
Really?
Oh. Wow.
This is beautiful.
Amanda, I think we saw one of these the other day, too.
Really?
I think so.
Yeah, we saw something similar to that.
Yeah, we did. We actually did because we were trying to figure out what that was. And it was at about this depth, too, I think.
A little bit. 19 delta.
Yeah.
70 degrees.
Wow. Oh, wow. Look at it.
Sparkle, sparkle. It's the 4th of July.
It is.
No wonder it's doing
These are your
this.
fireworks.
Right.
Okay,
so, Mike, have you made any progress in figuring out what these are?
78.
Ha.
Actually, I was going to say we did a reanalysis of some past midwater explorations, and it included an organism very similar to this, but it was clearly gelatinous. It included an organism very similar to this, but it was clearly a gelatinous animal with just many tentacles that were really fine. It had the same effect in the video. For a while, when we first saw it, we saw one of these before in Puerto Rico, and we thought it was possibly some sort of foraminiferan, and but we were debating back and forth around the country because those would normally be very small, and what we were looking at in the video was rather large. But on this recent reanalysis of a midwater exploration we did off of Hawaii, we saw a similar organism, but it was more clearly at the center a gelatinous animal, but it was highly reflective. So it was kind of difficult to really look at, but it might be something gelatinous, so it's certainly something we're going to have to stack up all the images that we have from all of these transects and dig deeper and see what it might be.
Mike, has anyone explored the possibility that these might be radiolarians?
I think we did talk that around too.
This transect to be at a 615. That'll be our endpoint.
I think it might have been a scale thing. If I remember the conversation that we had at the time, there was a lot of discussion on making sure that it was the right scale, that we're looking at the right size organism. So it's totally possible. I mean, all of those possibilities are still in play.
Yeah. I might know some people over in France. I've sailed with some radiolarian experts before. It might be worth sending along to that group just to see their thoughts on it.
Yeah, we totally should. At this point, we probably have at least three, with this one now, very good video sequences that we could send around to some experts to get their opinion. And kind of, write up some proper field notes on it.
I can come in partial and see. Oh, there's something center screen. It looks like a jellyfish. It's a ways away.
So you know that the gelatinous organism that we saw, wait, Annie's pointing something out to us. Wait, she's my spotter. Oh, looks like another hydromedusa. Oh, wow. You can actually see, I think you can.
It is a Siphonophore.
Is it?
Really? There's something, a jellyfish.
Yeah, that's a Siphonophore.
Huh.
And it's like, it's a rather different form. It's not quite lobate. It's almost completely spherical. This is great. This looks like Helicreus minimum.
Got you at 83.
This is a Hydromedusae.
All right, come on in video.
19 meter delta.
Sure. Oh, zoom.
Oh, wow. Ow.
All right, coming up.
Boy, hard to image those
maybe
guys.
Not. That's another,
There's something center.
I'm not sure about Helicreus.
Might be some.
But that's, certainly looks great.
What do you think, the gelatinous organism we saw early on in the 1200 meter transect that we weren't sure what it was?
2 minutes.
Could that have been a Typonophore?
Lower my lower swing arms.
Bring them out more.
The one with the long hanging tentacles?
No, we weren't sure. No, it looked, we weren't sure if it was a larvation
Top left
or...
The one that was kind of like doliolid. No, I don't know if that would be a Siphonophore.
I'll try.
If I were to get a Siphonophore, I would have to look for
those rows.
the home rows.
Yeah.
Those are pretty indicative, and you can find them on almost every Siphonophore.
Let's see.
So I know that we're not able to detect bioluminescence that well with, but are we? Did you see? I mean, to me, it looked like some of these things that we've been saying have been bioluminescent.
Yep, you are.
I got you at 83 degrees.
Some of them genuinely are bioluminescent, but a lot of what we might see is the reflection of the ROV light on some of the structures in the jellies, at least for the jellies.
Yeah, but when I'm in a submersible and we're going down in midwater, we have all the lights off.
Yeah, but our eyes, we have very sensitive eyes.
We do, but what I see is an iridescent bioluminescence, like on the legs of polychaetes.
Okay, what is this?
I have more zoom.
This looks like just
biogoo.
Yes,
down to keep you in view.
marine snot. Oh no!
Wait!
I take that back. I take that back. I'm so sorry.
Looking back at you.
Mike, what's this?
I'm so sorry, animal.
Amanda, what's
whatever
this?
it is.
Liz?
Time to come up.
Are we any more?
That's
no, that ends the transects.
Yeah, I think
All right.
this could be another
That ends the 500 and last transect.
Siphonophore?
Cool way.
Yeah, I was thinking Siphonophore, maybe Siphonophore parts.
Yeah, it looks like it might be missing its Metrophore.
So some of that stuff that's going to start settling down to the bottom that other things are going to be eating off of, and eventually bacteria will settle on.
Exactly.
Okay, that's it.
Let me know when you have any turn auto heading off.
That's great. By the way, that Siphonophore that we saw, the big one that was sort of like this ball. I think that might have been a juvenile form of a ctenophore called Thalassocalyx magnificus.
You see me.
There's a couple of these big ctenophores. One's Bathacoroe that has big lobes, and the other one, the Thalassocalyx, has more of a kind of a ball shape, less obvious lobes in the bottom.
Good.
But really cool stuff.
I want to turn auto heading off.
So before we wrap up, would it be possible to get each of you guys to summarize either your favorite thing or sort of an overall summary of what we saw would be, and sort of the value of what we did would be great for our videographers to capture.
And for our listeners at home.
And for our lovely listeners at home.
Sure, I can take a quick stab, and then I'll let Amanda and Liz and Shirley and others jump in. I think it was great we had a chance to do 6 levels today, starting at 1,200 meters and finishing up at 500 meters, where we thought we were in the deep scattering layer. I thought it was excellent. We saw a lot of Keegnap. We got to see a lot of plankton.
So I was talking with Dive Super, so I think if we could just keep what we're currently doing, looks like
another one of those mysterious, whether it's a foram or radiolarian or other
if we could keep doing that, that would be great right now.
I think some of the most exciting ones that I saw today probably was that last ctenophore that we saw. I think it was kind of neat to see that.
It'll go away as soon as we come out
of
position anyway.
That's kind of what we did. The value of it
sounds great.
is certainly the ability to come here to this place in the ocean that has not been explored in the midwater extensively at all, and this being our 4th midwater exploration in the Marianas Trench, when we were able to kind of broaden the frontier of what we understand about the midwater.
If you are not already.
So
thank you.
For me, that's pretty exciting.
I've got it up and running.
Ooh, thank you.
Well, I'll jump in and add to that a little bit. I thought the most exciting thing for me today was the fleeting possible view of the cephalopod in the camera 2. So I'm looking forward to going back and having a look at that. But my second favorite, my runner-up is the view of the shrimp, just because I think that it really showcases the extraordinary videography that is going on when you can get up so very close to see all the lumps and the bumps on the carapace and on the arms and on the legs. And it really was just a beautiful portrait of this animal, this tiny little animal in the middle of this very vast ocean. It's a great kind of icon of what's left to discover.
All right, this is Amanda. So some of my favorite things were that in some of the deeper transects, we saw a couple of organisms that none of us could even identify to, the phylum level. So the first one we saw was a string of, what looks to be some kind of gelatinous animal. They were sort of pumping like a jelly, but didn't quite look like a jelly, had no tentacles. The second one, again, was a gelatinous animal that looked a little scalpy, a little ctenophore with no teens. There was some coloration inside that we couldn't quite figure out what kind of organs that were. And it's just really amazing that, we can go out here and in just a couple hours see things we don't even recognize. And it was interesting to me that those were in some of the deeper depths, because the deep pelagic ocean is where we think that
Pilot Dive Super, comms check.
Most of the undescribed biodiversity likely exists because of how poorly explored that area is. So it's really exciting to get to be kind of at the forefront in this region and get down there and see what's there and learn about these animals.
Deck Dive Super, comms check.
I got you loud and clear.
Turn myself back on. Sako Matsumoto said that her favorite was the Hydromedusae when she commented cool. I think that might have been the one that had all those beautiful, amazing tentacles that actually reminded me of kind of like the Christmas decorations, those long flickering lights. Oh my gosh, really beautiful. Really absolutely beautiful. And lots of kudos to our ROV crew and videographers. This is a tough thing to do, to maintain this position in the water
yeah, so it looks pretty much about the same.
To try and
we have about
get that
0.66
camera
knots,
focused in
towards
and
the
to
west.
get this great imagery that we've gotten. So
maintaining,
really, we
bearing
are
at
so lucky to
110
have the high quality, the talent that we have of our ROV crew and our videographers. So thank you very much.
Okay, copy that.
It is. Okay, so Mike Ford, yeah, everyone's saying he has incredible midwater dive. All Mike Ford saying great dive, lots of great new clips to look at and more depth. Sign of a good dive in the right place. So thank you all. We really appreciate you joining us for this midwater portion of our dive over the Petite Spot volcano. And we hope to see you all tomorrow. We're going to another GEO called Geo, and it'll be a little bit shallower, about I think 2,000, 2,100 meters, something like that. So look forward to having you join us tomorrow for our dive, and because we're coming towards the end of leg 3 of this mission. So again, thanks very much. Thanks to all of our shore-based scientists for your participation as part of our science crew during this expedition, and we'll see you all tomorrow.
All right, thank you so much, Shirley and Patty.
Bye-bye.
Thanks a lot, guys. That was great.
Bye-bye.
Thank you. Good
You're
night.
You're welcome. Good night. Good night.
Bridge, seal back
Night,
there?
Liz, Asako, Debbie.
That's a negative bridge.
Amanda, Mike, everyone. Thank you.